Polymorphisms and human identification. A DNA polymorphism is a sequence difference compared to a reference standard that is present in at least 1 to 2% of a population. Polymorphisms can be single bases or thousands of bases. Polymorphisms may or may not have phenotypic effects. They're found throughout the genome. If the location of a polymorphic sequence is known, it can serve as a landmark or marker for locating other genes or genetic regions. Each polymorphic marker has different versions or alleles. There's different types of polymorphic DNA sequences. RFLPs, restriction fragment length polymorphism. VNTRs, variable number tandem repeats. STRs, short tandem repeat. SNPs, single nucleotide polymorphisms. So before we go into these different types and the details of them, why do we care? So the reason why we care is because if we want to investigate a crime scene and compare DNA from a known criminal to the crime scene, or if we want to determine paternity, it's really not feasible and really unlikely that we would try whole genome sequencing. Now, whole genome sequencing is becoming more and more readily available, and it's becoming a lot easier to complete. But still, we can't get around the fact that whole genome sequencing requires a whole bunch of storage and data. So instead of sequencing the entire amount of DNA that's in front of us, we typically just evaluate particular regions. And if we can amplify particular regions, those polymorphic regions, then we can compare them and just look at those individual or independent regions of DNA to do our comparisons. So that's kind of what this is focusing on, is really like identifying crime scene, the people who are responsible, words are escaping me right now, the people who are guilty of committing particular crimes, or the victims' DNA at a crime scene, and paternity testing. We can also use these to identify particular genes or differences in genes, but in general, a lot of this is going to focus on like forensic sciences. So restriction fragment sizes are altered by changes in or between enzyme recognition sites. So we talked about restriction enzymes, and restriction enzymes are going to recognize, they're going to read the DNA, and they're going to recognize a particular sequence and cut at that sequence. Well, if there is a difference in that sequence, it may cut in a different spot. The presence of an RFLP is inferred from changes in fragment sizes. So RFLP genes or genotypes are inherited. For each locus, one allele is inherited from each parent. So here we can see we've got the father and the mother and the child. So if we look at the child, we can see that these two bands came from the mom because they're in the same size. Now we're looking at where these two bands may have came from. So if we look at these on the father, that's where they came from on the father. So all of the baby's bands have to be part of either the father or the mother. Which alleged father's genotype has the paternal alleles? Well, in real life, these aren't going to be color-coded. So you would actually just go through them, and you would start by looking at the child's, and then you want to account for all of the bands by both parents. Now, typically, we know the maternity. We know who the mom is of the child. So, because we're like pulling the child out of the mom, so we can typically know that that's the child's mother. So we go down the bands and we start reading these. And we're like, oh, well, this one came from mom, so we're not going to worry about that one. And we're going to keep going. And then we're going to say, okay, well, this one came from mom, so we're not going to worry about that one. So now these two bands would have had to come from dad. So which one of these two dads has those two bands? Well, the only father that's possible is this second one right here, because these two bands would have come from there. And that's really how they would evaluate RFLPs for paternity testing. Evidence testing by RFLP. So which suspect, 1 or 2, was at the crime scene? So here's suspect 1. Here's the parent of the child victim. Here's the child victim. And here is the suspect number 2 and crime scene evidence. So when we look at these two different gels, we're going to go through and evaluate the child or the evidence and like where this evidence would have come from. So if we look here, the evidence here shows 2 bands, and the only individuals out of everyone else present that has those 2 bands is criminal number 2. Looking at the evidence here, we go down, and here's the evidence. The only criminal or the only other individual that has those 2 bands is again criminal number 2 or suspect 2. So suspect 2 now has a high probability of being the one that actually had their genetic material at the crime scene. Short tandem repeats. STRs are repeats of nucleotide sequences. A mononucleotide would be just 1 letter that's repeated. Dinucleotide would be 2 repeated. Tri would be 3. Tetra would be 4. Penta would be 5. Different alleles contain different numbers of repeats. So 4 repeat alleles are present here. 5 repeat alleles have 5 sections that were repeated. Forensic scientists analyze the number of repeats at specific STR loci, locations in the genome. By comparing the STR profiles of DNA samples from a crime scene with those of suspects, scientists can determine if the samples match. Again, we don't have the capacity to do whole genome sequencing and analyze all of that data and store all that data. So instead, we just look at particular locations in the genome. This allows for the identification or exclusion of individuals in forensic investigations. STR alleles can be analyzed by amplicon size, polymerase chain reaction. So we make many copies of a particular region and evaluate it from there. Allelic ladders are standards representing all the alleles observed in a population. So here's the PCR products that were produced. Allele 1, 187 base pairs, 7 repeats. Allele 2, 191 base pairs, 8 repeats. Multiple loci are genotyped in the same region using multiplexed PCR. Allelic ladders must not overlap in the same reaction. The amylogenin locus is not an STR. The humamyl gene locates or codes for amylogenin, like protein. The gene is located at XP 22.1 to 22.3 and Y. So, the P arm of the X chromosome, band 22.1 to 22.3, and the Y chromosome. So the locus, females have 2 X's, the males have XY. STR genotypes can then be analyzed using gel or capillary electrophoresis. STR genotypes are inherited. 1 allele is inherited from each parent. Which alleged father, 1 or 2, has the child's paternal alleles? So here's the child. The child is 16, 17. It got the 16 from mom, so it would have to get the 17 from dad. Here we have a 14, 18. Well, it got the 18 from the mom, so it would have to get the 14 from father number 1. Here we have 21, 24. The 21 came from mom, so we would have to pick up the 24 from the dad. Well, both of the dads have that. Here we just have a 6, and all of the involved individuals have the 6. Here we have 10, 11. We've got the potential for either the 10 or the 11 to be coming from mom. But here we have an 11 coming from the dad, and there's no 10 or 11 on the other ones. So again, more evidence that number 1 is the father. And then we kind of go through here. 11, 12, well, 12 came from the mom, so the 11 could have come from either dad. 11, 13, the 11 came from the mom, so the 13 could have come from either of the dads. And 9, 12, the 9 came from the mom, so the 12 could have come from either one of the dads. So evaluating all of these different regions is necessary because we want more than one piece of evidence that that's the father. And now we have 2 out of these that confirms that that's the father. Forensic testing by STR-PCR. In 1997, the FBI adopted 13 core loci as the Combined DNA Indexing System, CODIS. The genotype or profile of a sample is the collection of alleles in all the loci tested. Genetic concordance, all locus genotypes or alleles from 2 sources, for example, the suspect and the crime evidence, are the same. Concordance is interpreted as inclusion of a single individual as the donor of both genotypes. So matching the probability of STR genotypes in subpopulations, you can see the more loci you use, the stronger likelihood that there's going to be a match. STR polymorphisms, the Y STR. The Y chromosome is inherited from father to son without recombination. STRs on the Y chromosome are therefore inherited paternally as a haplotype. Y haplotypes are used for exclusion and paternal lineage analysis. Mitochondrial DNA can only come from the mom. Sequence differences in the hypervariable regions of the mitochondrial genome are depicted below. Mitochondria, again, are maternally inherited because if you think about what contributes to making the baby, you have the sperm, which is super tiny, doesn't contain any mitochondria. It's just genetic material encapsulated with a motor protein that's going to allow it to swim. The egg has all of the organelles. So every individual will only inherit their mitochondria from their mother. So they're maternally inherited. There are an average of 8.5 base differences in mitochondrial HV sequences of unrelated individuals. All maternal relatives will have the same mitochondrial sequences. Mitochondrial typing can be used for legal exclusion of individuals or confirmation of maternal lineage. Single nucleotide differences among DNA sequences. One SNP occurs for about every 1,000 bases or base pairs in human DNA. SNPs are detected by sequencing, milk curve analysis, or other methods, and we talked about those methods previously. 99% have no biological effect. 60,000 are within genes. SNP haplotypes, they're inherited in blocks or haplotypes. SNPs can be used for gene mapping, human identification, chimerism analysis, and many other applications. The Human Haplotype Mapping HapMap project identified SNP haplotypes through the human genome. Engraftment testing using STR. Allogenic bone marrow transplants are effective treatments for cancer and diseases of the immune system. Bone marrow transplants are monitored using STR. So, bone marrow cells come from within the individual and they are autogenic or autologous. But donated bone marrow through a bone marrow transplant are allogenic. A recipient donor or a recipient with donor marrow is a chimera. It's kind of a combination between the two. STRs are scanned to find informative loci, donor alleles that differ from recipient alleles. There are different degrees of informativeness. With the most informative loci, recipient bands or peaks do not overlap. Stutter in donor bands or peaks should be avoided as well. Stutter is a technical artifact of the PCR reaction in which a minor product of N1 repeat units is produced. And then you can review the summary.